Hello, everyone, and welcome to today's episode of our speaker series as part of our symposium. Today's episode is very special to me because this marks an occasion that many people thought would never come in our arduous journey towards gene therapy. We are, in fact, in contract with biotech. This biotech is the Charles River Labs, and they are a biotech plus a CRO, a contract research organization. So they bring to the table a lot more than a normal biotech in the sense that they have a lot more experience with a lot more gene therapy trials. Today, we're going to be speaking with Dr. Keith Sutton, who is their scientific advisor. And his job is to work with clients to get therapies to clinical trials ASAP. Dr. Keith Sutton is working with us directly on taking over and working with the mouse model and expediting things for us. So I will not delay the conversation any further. I will turn it over to Keith and request that you put your questions, if any, into the chat box or in the Q&A tab. And once the presentation is over, we will visit those and address those for you. So Keith, I'm going to turn it over to you, and thank you in advance for making time to make this presentation for us.
No, thank you very much. I mean, it's really exciting to start working with you as you look to bring your gene therapy to your patients, to your community. And today I'm going to talk about, in general terms, demystifying the gene therapy process. So I'm not really going to specifically talk about the program, which we're just initiating, but more a kind of a broad overview to thinking about how this works, how we get from an idea, a concept, to what we want, an effective and a safe treatment delivered to patients. So first of all, just maybe a little about Charles River, or just actually an overview of what I'm going to cover in the talk. So first of all, just an overview and a roadmap of getting a gene therapy to the clinic. We're going to talk a little bit about gene therapy approaches, AAV approaches in particular, and that in many respects, we maybe think of these as a new approach, but we have a huge amount of knowledge about the challenges and potential liabilities that can impact the development of these programs. I'm then going to talk a little bit about the regulatory phase, actually working with the FDA. And actually, I would say you do work with the FDA if you really want to get things to work as quickly and smoothly as possible. And I'm going to also talk a bit about one of the challenges that does exist for gene therapies that can sometimes be overlooked. And that's actually about managing the kind of the quality attributes, actually making sure that the therapy is safe and has an ongoing safety profile, obviously, as you're progressing in the clinic. So just about Charles River, I think as Lally said, we're a global organization, I think now well over 10,000 employees. We're as a main activity as a contract research organization, and we do not kind of, if you like, generate the de novo intellectual property for the therapeutics. Our role is to take those ideas, and those ideas come in at different levels. We work with large pharma companies, virtual biotechs, patient-driven advocacy groups. And it's all about getting at each stage as quickly as possible. So I suppose in terms of the current discussion, it's actually getting from that early stage concept, developing the proof of concept. Is there a therapeutic approach? Is it viable? Getting into safety assessment. And this is where you start the interactions with the FDA to say, this is now the path to the clinic. And then we can also offer manufacturing and the CMDO CMC support for AAV therapies. So myself, I'm actually based up here in the UK, based out of the Edinburgh site, but work actually quite broadly with US companies and with them navigating the IND process. So gene therapies, and I think most of us heard a lot about gene therapies. It's in the news nowadays. Every other month there's some news about a new gene therapy approach and the potential of that, or how it's performing in the clinic. And for gene therapies, what we're really doing is we're taking advantage of the biology of viruses. So viruses have an incredible potential to travel around our bodies, to target cells. And when we're thinking about a gene therapy approach, when we start thinking about the viruses that are available to us, and we strip out, we re-engineer them, we rebuild them. So they're no longer an infective virus, but rather they're a tool for the therapeutic delivery of a payload, a gene therapy. So replacing something that is missing within a patient group. But we're taking advantage of the basic biology of the virus to deliver this goal. And that has advantages because we're dealing with a normal biological structure. So those viruses, when we select the virus, we know which cell types it will preferentially deliver its therapeutic load to, so we can select a virus that will hit skeletal muscle and so deliver to the right place. So thinking about what's the translational journey for a gene therapy, what are the steps and stages that you go through from an idea, a concept, as you're looking to get this as an idea that can get into the clinic and get to a patient population. So what's that translational journey? So translating that basic insight into a therapeutic. So it always starts back here with the idea of a therapeutic concept and understanding the biology and the pathobiology of a disorder. And in that case, we're thinking about, well, what from that is the therapeutic rationale? So here we're thinking about looking at a gene replacement approach to provide a functional copy that can fix cells which are being damaged by the lack of that enzyme. And that kind of leads us to the next step, which is identifying a clinically viable strategy. And I think one of the important things to emphasize, even very early in the process, is the goal is a clinical product. So we're really focused in on something, on an idea, on a concept that can be delivered through this entire process into the clinic. So the next step then is the proof of concept. So we need to start building out the gene therapy construct, the AAV construct carrying the therapeutic gene. We need to think about how we test those candidates. And this is to demonstrate, if you like, drug on target. I mean, I personally, sometimes people talk about this as efficacy. I'm a bit reticent to talk about efficacy because invariably these are mouse models often we're working on. And so really what we're looking here is a confirmation of the therapeutic approach. And that allows in a process of refinement to select out what will be the candidate, the actual drug we're going to look to deliver. So we might start the proof of concept phase looking at a number of different possible constructs. Those can be optimized, they can be tweaked and tuned, so we're getting the best indication of a potential efficacious response in a patient. And at that stage, and I think this is the bit I wanted to touch on as I go through my talk, the nature of the process shifts. And so this is the change in the process. And this is sometimes the bit when you hear from academic groups, they'll talk about, or you'll read an article about identifying a gene involved in a process, and they'll then say, but it will take us however many years, 5 years, 10 years to get to clinic. And one of the step in here, and this is the phase which I think we'd really work on at Charles River, we really work on to focus in on and to concentrate and to streamline. And that's actually the safety assessment phase, because the goal or the primary activity of the FDA, or in Europe, the EMA, is about safety. Their job is to make sure that patients are taken care of. And in the safety assessment phase, that is what we have to do. We have to generate a program of work which is going to convince the FDA that the gene therapy is safe. And so that involves thinking about a regulatory strategy to reach the IND, and that involves interaction and discussion with the FDA and a feedback from them. And from experience, we can actually shape that out quite early, and I'll touch on this later, what that program of work looks like. And from that work, out of that work, we get the IND, we get the permission to dose the first patient. And so this is really about setting and identifying a starting clinical dose. And then we get into the part, which I think everybody wants to reach, and we want to get to, which is actually getting into phase 1/2, and clinical trials. And that's where we start looking at what's going to be a clinically relevant dose. Again, the real goal, the focus is safety. And also, of course, now that we're in patients, now we can talk about efficacy. Is there a clinical benefit? And once you're in that process, moving through the clinical phases, you're again talking with the FDA and hopefully starting to move towards a BLA and actually a licensed medicine. And at this stage, obviously, I mean, looking at probably to partner with a larger organization, but that's often very easy, but it's much simpler when you've got a clear, efficacious therapeutic with demonstrated clinical attributes. And then the other thing which I wanted to really touch on is one of the challenges for gene therapies is that they are complex biologics. Viruses are not simple structures, and the manufacturing of these to a suitable standard and quality is one of those areas which can sometimes trip up and slow down smaller, less experienced organizations, because the timescales here are quite long in order to get this process working and to have that kind of control of the drug product satisfactory for the regulatory agencies. Pardon me. Okay. So thinking again about that very first stage of the concept. So again, at this point, I'm going to talk quite broadly, not really very narrowly focused on our current work with you guys, but thinking about gene therapy and getting from concept to proof of concept. So I was going to start off maybe just chatting, outlining a bit, and maybe some of this, I apologize if you've already heard talking about AAVs and viral vectors. So when we think about virally vector gene therapy, we're actually starting with a range, and not just AAVs, which is mainly what I'm going to talk about today, and for the program we're supporting, but there are others, alpha viruses, the lentiviruses, also herpes simplex viruses. One of the features, now these all differ in terms of their biology and their structure, but one of the things that's common to all of the parental organisms that we use for gene therapy approaches is their persistence within cells once they enter a cell. So in the case of the AAV viruses, the genomes can catamerize, they link together within the nucleus, and they become very stable in that format. And this has the advantage that you can get years of therapeutic benefit. I think in many of the ongoing trials that are continuing to be published for some of the earlier gene therapies, efficacy is being seen 3+, 5+ years after dosing. And so that's a real, gene therapy does seem to be showing that long-term promise that these are not going to be transient therapeutic benefits. And again, this question of tropism. So when we talk about tropism in biology, we think about which cells the virus interacts with and can deliver its payload to. So the adeno-associated viruses are the most commonly used and frequently for programs looking for muscle delivery. Actually, the serotype that's used is AAV9. But there are 13 kind of parental characterized AAV serotype, and each serotype has slight differences in tropism based on the structure and shape. So there's actually a 3D kind of rendering of the kind of protein capsid. These are non-envelope viruses, so they are a naked protein complex that interacts and passes into the cells when they transduce and introduce the therapeutic cargo. And the capsid structure of the AAVs is very well understood, and there's a lot of potential ongoing for many companies looking at re-engineering capsids. Although the advantage is that for the ones we know, muscle tropism is quite well established. And I think one issue that does, or as often comes up when we think about gene therapies, is of course these are viral vectors. They are viruses that are present within the population. But the actual prevalence of seropositive individuals is acceptable. It can be quite low, and there's been some recent work saying that even in cases where there is some evidence of prior exposure and immunity to a particular serotype, that doesn't seem to greatly impact therapeutic benefit. So it's not, doesn't look as if you need to be truly negative for any prior exposure. And AAVs, including the AAV9s, are well established in the clinic. And we understand, and I think this is an important thing, what some of the risks and liabilities are of gene therapy approaches and what the challenges are around addressing the safety question. So what do we need to think about, both from a basic biology point of view and also from clinical risk? And again, when we're thinking of working with the FDA to get into the clinic, we've got to think about what are anticipated or potential clinical risks that we need to think about, that we've looked at them, we've got enough data to reassure, well, actually, let's be honest, we've got to reassure ourselves. Nobody wants to develop a drug that isn't fully safe, and to also persuade the FDA. So challenges and liabilities for gene therapies about, so what do we know, what's been established as some of these issues? So the first one doesn't really apply, fortunately, to the current work. And one is actually the amount of material you can get into, the size of the gene you can put into an AAV is quite limited. It's about 4.7 kb or less than that. And I would just say, and I've got a slide after this to talk about, you can manage that, and that can be managed and mitigated. The other challenge is that the tropism of the AAVs is not precise. It's not exact. You don't only hit skeletal muscle, and you don't only hit the liver or the heart. These are viruses. They have preferential tropisms, not absolute tropisms. So we need to be very careful about thinking about, we have a target population of cells we want to deliver our therapy to, but we'd also have to be aware that there are other cells in the body that are also going to take up that TARC transgene. And those cells may not be a good place to express that therapeutic. And this is one of those aspects that we look for and we assess, and actually during the early, that kind of proof of concept phase. And again, these are viruses. So we need to think about immunogenicity, the immune response to the therapeutic. Now, one advantage here of using viruses is a lot of viruses, including the AAVs, are actually very good at evading the immune system. And so they're not terribly immunogenic, but we need to understand the impacts. That is in terms of antibody response, humoral immunity that might prevent the therapeutic being delivered to the target cell population. It can also be the cell-mediated immunity, so the arm of the immune system that kills cells that express foreign non-self proteins. And so we need to think about what that impact is. How do we assess that? How do we reassure ourselves that that's not going to have a negative impact on the development process? And importantly, as we've worked, as many companies have worked and run programs looking at gene therapy vectors, there are some particular sensitivities and concerns. So the liver is the main source of, so if you use an intravenous administration as you would to reach skeletal muscle, the liver is actually the main organ that is hit. The role of the liver is to take and to filter, purify the blood. And that does mean that a lot of the AAV is going to end up in the liver, as the liver functioning as a sink tissue. And that can have impacts on liver function. And many of those aspects can be well managed within the clinic, but it is a question and a concern that always needs to be looked at and addressed during the development. And then there also seem to be other cell types, and the main one for AAVs is the dorsal root ganglia. These are neuronal structures associated with the spinal column. And at very high doses of AAV, these neurons seem to be particularly sensitive to perturbations. And it seems to be more around transgene expression and translation in these cells rather than a stress response to actual viral infection or to an immune response against those cells. So these are the sorts of things and the areas where the regulatory agencies want us to look and consider and think to show that what we're developing is safe. So actually, I wanted to just put this up because I thought, in terms of actually maybe just also to mention a success story that's going on actually at the moment. And so if we think about many of the genes for other genetic conditions that we want to target, they're actually too big to fit in AAVs. The main one obviously is dystrophin. That gene is 14 kilobases long. I mean, I think this is a little kind of figure from a paper from Dongxing Guan. And the solution that people have come up with in that field is to engineer and to explore, if you like, what's the minimal structure for a functional dystrophin protein, so you can get it into an AAV and deliver it to the muscle cells. And actually Pfizer have currently, I won't read the number, a mini dystrophin in phase 3 for Duchenne. And that seems to be progressing well from the bits of data that they've been releasing publicly from that program. So the other challenge which mentioned was this bit that the tropisms, the cell types impacted that take up the therapeutic protein are not always the cell types where we want function. And the general, one of the simplest ways of managing this process is to use tissue-specific promoters. So for example, muscle-specific promoters if the therapeutic is being designed to deliver or to replace defects in muscle function. And so we can look to use a specific promoter to ensure that we don't see, for example, expression within the liver or within the dorsal root ganglia. So these are ways of mitigating, of attempting to limit impacts on non-target cell lineages. However, that gets you so far, and I think there's been an interesting story with the limb-girdle muscular dystrophy and the calpain-3 application. And I think I bring this up just as an example of what you might think of was a dead end in a program, because the challenge here is that if you express calpain-3 within the heart, you end up with fibrosis. So it's not a viable approach. But what Ruda and colleagues did, and actually it's a beautiful piece of work, it's a lovely paper, is that they took advantage of an endogenous pathway within the heart, microRNAs, to downregulate that transgene. So you can now have a strategy where you can't, at the promoter level, differentiate between the heart and the skeletal muscle, but by taking advantage of these specific microRNAs in the heart, you ensure that the transgene is not expressed in the heart and the cardiac muscle, and so you don't see the damage there, but you still see the potential or have the potential therapeutic benefit within skeletal muscle. And so this is just a bit about thinking that we have these challenges. Some of them we can anticipate, some of them will be unknown, and I think but there are biological processes and solutions that we can always look to deploy and consider to get us through and to make sure we keep on that plan to get to the clinic. So there are challenges in the proof of concept phase. That may be around the structure, the challenges of viral delivery and targeting the therapeutic gene and limiting expression of a therapeutic protein. But there are model challenges. So often, if we want to show the potential for clinical benefit, we need a model, especially for genetic diseases. The most common model used is the mouse. There are exceptions. So for example, there are pig models of Huntington's, and there's obviously canine models of Duchenne, but for most situations, we have to make use of mouse models. And that is, at one level, an advantage, because mice are very genetically amenable to these sorts of disruptions, so we can find or generate a specific model for a given disease or indication. But when we come, and this is, I think, why we sometimes always kind of get a bit resistant to talk about efficacy in this stage, because the pathobiology in the mouse is often quite different from what we see in people, and that's hardly a surprise. And that can be differences in severity. Sometimes we see quite profound differences in the organs affected for a given genetic modification, and the time course. So sometimes we have mouse models that you might need to wait for 12 months before there's any pathobiology developed. And that presents challenges, and we need to think about how we meet that and how we address those challenges. And so one of the things when we're thinking about these models and how we use these models is, I think, avoiding getting trapped in feeling the need to prove efficacy. And this is the idea of capturing, this idea of making a distinction between capturing proof of concept and not efficacy. We really want to show, and I think the phrase people often use it as drug on target. Do we see the delivery of a gene to where we want it to be? Hopefully we have a mouse model where we see some physiological, pharmacological response in that animal that indicates we've got a drug on target. It is replacing, giving a functional replacement to the ablated gene, the genetically modified mouse. And the other thing that these early models can provide, which again is really important, just thinking back to what I was talking about in the previous slide, is they give you the very early readouts of unanticipated toxicities. I think a common question we always get when we're talking with clients around gene therapy programs is, what's a standard gene therapy program? And part of the challenge of gene therapy programs is they're not standard. They're always slightly different. The nature of the gene, the potential issues about overexpression of a gene in cell types where you don't want that overexpression can lead to challenges and complexities. And so you want to make sure that you're always looking at this kind of live and considering, and really thinking hard about your no-go decisions, about what is it you need to see. And when you're planning these experiments out, by and large, experiments should have quite narrow goals to demonstrate key attributes. So do we see delivery to the right cell types? Do we see production of the therapeutic protein? Do we see drug on target? Do we see any evidence that this is actually giving us some clue towards an indication towards clinical efficacy? And so this is this whole question about demonstrating the desired pharmacology. And so in this early stage, this is what you want to focus in on. Do you have confidence that the pharmacology is right? Now, the biology of the disease, that may be absent because of the limitations of what we're working with models, with mouse models. But these experiments do provide us with this initial confidence that we've got drug on target. And they can often give us very good, solid kind of no-go thinking, that we don't have really unanticipated adverse events or toxicities based on sometimes expressing too much of a transgene can lead to cell damage, even if it's in the right cell type. Other times it's in inappropriate cell types. And so these models can give us that very quick readout about whether there's something here we hadn't considered. And that's really critical in the drug development process. You know, bad data early is good because it saves you a lot of time and money, and it tells you, right, we need to stop, we need to take a step back before we go forward again. You know, if you've got a bad drug, there's nothing you can do to make it good. I mean, that's the real concern. If it's not working, you need to stop. Don't keep it alive. Don't try and keep it going. Reconsider things, because often we're not as smart as we think we are. There are aspects of biology which we just didn't quite understand as well as we thought we did. And then we need to think about where this fits within this process. So one of the things to really remember when we're thinking, because now we're thinking, okay, so we've got through our proof of concept studies, we've got a candidate. we've got something which we're confident is on target, doesn't have real unanticipated impacts on the physiology of the organism. It's going to be safe. We've got confidence it will be safe. We've got the evidence there that it's likely to be efficacious. We've got to remember that these studies do fall into the information we put in front of the FDA. And the FDA, when they look at this information, this kind of pharmacology information, that they're not looking for you to prove that you will have efficacy in the patient population. They want to have confidence that there's a likelihood that that's going to have clinical benefit. You have drug on target, you express in the right places, you have clear evidence of protein production, it's doing what it needs to do. So all the evidence is there that in a patient, this has a good chance of providing clinical benefit, like many other gene therapies have already. So we've got to think about where this is. But the other thing that often the regulators will do, especially for gene therapies, is they will look through some of the kind of the pharmacology data to maybe get clues about, is there anything here? And this is why it's important that when we develop a drug, we test it ourselves. It is important to make those key assessments so so that you can make that presentation to the FDA, to the regulators, that we have confidence that this is safe. And one thing actually to emphasize here and actually to bring this up is that the complexities of gene therapy manufacture and the kind of requirements for the safety assessment phase, which is what I'll kind of move on to touch on next, is that it's actually almost at this stage that the big decisions need to be made about moving towards a strategy for manufacture and production of a clinical material. Okay, and this is one of those kind of particular challenges of, I mean, to be blunt, quite financial demands of developing a gene therapy. You need to have your clinical material or your clinical equivalent material ready for your safety assessment studies. And so that takes us nicely to this last kind of part. Okay, so getting from proof of concept to the clinic. So what happens? I mean, this tends to be the black box, I think, that kind of disappears from public sight. We hear, as I said, there'll be announcements, the gene involved in a certain disorder has been identified, and that's a candidate for a gene therapy. And people will maybe, there'll be a publication saying in the mouse genetic models, they get evidence of a therapeutic response. And then it kind of disappears from kind of public view for, well, hopefully for a short period of time. And so this is what I really want to touch on now. What happens when that kind of disappears? Okay, and this is the safety assessment phase. So thought I'd maybe just give a kind of high-level summary of this process. Maybe there'll be some questions later. I'll have to answer some questions. So the goal here, what we're working towards in the safety phase, is an IND authorization, permission from the FDA to treat patients with your therapeutic. And that authorization is granted by the FDA on the basis of their assessment of the data which the drug developer presents to them. Okay, so that's the job. That assessment is the critical job of the FDA, is something safe. And that's really what they want to know. The gene therapies are actually regulated, so there are two main parts of the FDA for kind of drug approvals and IND approvals: CBER, who cover biologics, and CDER, who cover small molecules, so the aspirins and the ibuprofens and those sorts of drugs, small molecule drugs. And they're quite different, and the regulations that feed into the assessments that need to be formed are quite different. So for biological drugs, because they're derived from, they are biological entities, they're made up of protein and nucleic acid, there are lots of liabilities that don't exist in the same way they do for a small drug. So we don't need to think about genotoxicity assessments. There's less concern about some of the safety pharmacology assessments that you need to do. So at one level, there's a lot less kind of peripheral concern about unanticipated consequence of that drug, because we know a protein or a gene therapy is just broken down to amino acids in the body. There's no worry about what those little bits that are left might do. And one key thing about any interaction with the FDA, and I think to really emphasize, CBER will provide opinions when you consult with them in the pre-IND stage and a proposed strategy to reach an IND. And I think a lot of the mistake people sometimes make, that the job of CBER is to assess what you put in front of them. It's not to design a program, okay? So never ask the FDA or CBER, what should I do? It's your drug, and their attitude is it is your drug, you are developing it, you should know what the concerns are, what the safety liabilities are actually going to be or might be, how have those been addressed in your program? And actually the main source where you get that critical feedback from this CBER is at the pre-IND meeting stage. And so this is a meeting you will have when everything is in place. And it's actually important to assure ahead of that pre-IND meeting, everything is in place. And you have a very clear program. This is what we're going to do to make the drug. This is what we're going to manufacture it. This is how we're going to test it safe, okay? The safety assessment phase. And then the clinical part of that exists as well. This is what the clinical trial is going to look like. And within that, as you're developing that drug, you're thinking about what your drug is, what your manufacturing process is, how you're going to show that each time you produce the drug, it's the same drug and it's always going to have the same properties. How are you assessing it? How are you going to show it's safe? Are you going to demonstrate what are the risks that you've identified? How are you going to capture those risks? How are you going to show that they're absent or manageable in a clinical setting? And what's really clear, and I think this is when the aspect comes down to working with the FDA. If you provide the FDA with a very clear, well-structured proposal about what you're going to do, why you're going to do it, why you're not going to do some other things, if you give them a clear plan, they will provide very clear feedback on that plan. If the plan is acceptable, they will tell you it's acceptable, or it's likely to be acceptable, because obviously data depending. But if there are gaps and they think, well, no, we think you should do this, they'll tell you. And there is actually scope to have some to and fro with the FDA, with CBER, about exactly what a proposal should finally look like. But you need to have a very clear idea as you're developing your drug. What is your drug? How are you going to put it together? What are you going to capture in the plan? Okay. So we talk about these kind of in terms of art in comments as pivotal studies, safety studies, IND enabling studies. And so this is the critical thing. And the role of these studies is to demonstrate safety. Okay, so we don't in these studies tend to look particularly at pharmacology. Sometimes we do. Sometimes we have to use models which incorporate the disease state, because that may have impacts on how we assess safety. For gene therapies, the basic assumption is that a safety study will last 6 months. And this is largely based on the fact if you dose a gene therapy, that benefit, well, that therapeutic is going to sit in the patient 5, 10 years. We're not entirely sure at this point quite what that end stage is. So it's going to be there a long time. So there's a reasonably high bar there in terms of how long you need to look and monitor to ensure that that drug is safe. And it's important to remember that these studies must align with the clinical regime. So what are you going to do within the clinical trial? So IND enabling studies exist to support a defined clinical regime. What are you going to do in the clinic? Then you can define what you're going to do to demonstrate safety, because that's the context you're demonstrating safety in. This is this patient population. This is who we need to show it's safe in. And so you build something to show it should be safe in that population. And one of the key things is dose levels. I mean, for any drug, there's a question about what is the level of that drug that's safe. But at some point, most drugs become toxic, and that's true of biologics as it's true of small molecules. Things start happening which aren't good. You can't keep pushing the dose indefinitely. And then the other key thing, especially for complex biologics, the material we use in a safety study must be equivalent, because this is what we're testing. We're testing the safety of a final drug product. So what we test in safety assessment has to match, and we have to be able to demonstrate it matches what we use in the clinic. It's a key step. And this all gets hashed out at the pre-IND stage. So we go in and we think about CMC, The manufacturing process, how we're going to manage that. Safety assessments, you would present to the agency. These are the studies we're going to propose. You're obviously also going to include your pharmacology or proof of concept data to indicate clinical benefit, evidence for clinical benefit, and then the clinical plan. And again, everything references to each other. Okay, so when we're thinking about how we design these studies, the key driver is this: it's the clinical population. Safety studies are driven by clinical use. And then we're thinking about, well, what's the AAV serotype? Where does it go? How long does it last? About the payload, immunogenicity, the level of expression, the pharmacology. Do we have any evidence of it doing what we're going to do? And then we need to think about toxicological risk. So this is what the agency is particularly interested in. If we have known liabilities, how are we checking those aren't there, or we've mitigated them in some way by design of our vector and our construct? And then there may be specific unintended pharmacology, because expressing our therapeutic protein in the wrong place. And again, toxicity related to dose. How much? Are we getting too much? Is that going to be a problem? Where do we set that bar when we come to think about patients receiving a dose? Okay, so just to finish off, and I think that's fine, probably some time for questions. So one thing to really touch on, and I think it is an important consideration when we're stepping back and thinking about the totality of a gene therapy program, and especially if the goal is to be as quick as possible. Because when we're working with gene therapies, these are, as it said, highly complex biological structures. Okay, so this is aspirin up here, just by a comparison. And CMC exists for aspirin. Okay, the FDA need to know if I sell a pill and I say it's an aspirin, how do I show that's aspirin? But from an analytical point of view, that's dead easy. Okay, it's not terribly difficult for me to take that material, put it through a mass spec, put it through all sorts of kind of analytical, not that many analytical approaches, and show exactly what I have in terms of chemical structure. I can show there are no degradation products. I can show there are no kind of chiral forms. I can do all of these sorts of analysis, and I can very easily specify my material because it's a simple organic compound. And the aim and the requirement here is actually a really obvious one from an FDA, from a regulator's point of view. So you've run safety studies, you run pharmacology studies, and they've looked at that data and they said this drug is safe. You can put it into people. Okay, so how do we show that the next time we produce that drug is still safe for people? It's the same material that we've shown is safe. And for gene therapies, that's actually really not as simple as it is for aspirin. I mean, the AAV capsid, okay, only has 3 proteins, but the molecular weight is 4 megadaltons, apparently. I mean, we know quite well what the structure is and the number of copies. So it's a huge structure to define and characterize. And so we can't define and characterize it the same way we do aspirin. That's not going to work. And it's also this issue that the manufacturing process is complex. We need to use cell lines to produce these vectors, these therapeutics. It's a very complex, it's a biological production process. And there are challenges because it's a complex biological process we use to generate the therapeutic. We don't get a fully uniform product out at the end. So we see empty capsids, partially full capsids, and these need to be characterized, these need to be mapped out. And then there's complex post-production and also the handling and the storage of the material. This is a biological material. You have to store it appropriately. If you store it inappropriately, it will lose activity. And so this really is just to say that when you're looking at the process, you need to think and plan for this kind of CMC aspect early. And otherwise it slows everything down. If you wait until the end, if you like, and if you regard the process as proof of concept, safety, and they say, okay, we're going to start CMC here, the CMC has to be complete before you can start your safety studies. And that CMC process can take 6 months, it can take 12 months. And so it's not straightforward to get, because now we're talking about manufacturing the clinical material, the equivalent material. So within that kind of context of program management for developing of a gene therapy, actually, one of the more complex, one of the more challenging aspects is building in this strategy to manage the CMC process, and that it needs to be in place before you begin safety assessment studies. And that does mean, again, thinking about decision points within gene therapy development, they tend to come, it's important that you're making them very early, because you get into a lot of moving parts that need to be initiated and commitments that need to be made. So it's thinking about when you're thinking early on in the process, how are you making and mapping your go, no go decisions? Because if you can make them early, you can keep moving, you can stay on track for that rapid development. If you don't have them, or if you have challenges in terms of models and they become difficult to make, everything actually ends up having to slow down, because you can't run things. You have to run things, or you get trapped into running things sequentially. Okay, so that was, I apologize, something of a lightning tour, and hopefully I did something towards demystifying rather than adding more layers of confusion, hopefully. But in terms of the takeaways, one is to say that gene therapies are now quite a standard therapeutic approach. We understand them, we understand the challenges, from a drug, purely thinking now about the conceptual, from the drug development stage, and that idea of getting to that concept. So we know what they should look like, especially when we're doing a kind of enzyme replacement, protein replacement approach. And challenges will come up in that process, but we understand those. I mean, most of the time, if a problem comes up, it's something which has happened in other programs or with other disorders, and we can think about, well, how do we mitigate against that? You need the decision-making points. You need, and I think this is actually broadly true of any sort of drug development process, you need to be ruthless. It's about getting a therapeutic to the clinic. If you keep a program hobbling on in the hope it will fix itself, they never do, and you're just wasting time and money. So you've got to be brutal, you've got to have decision-making points, you've got to go, no go. And those are challenging decisions to make, and it is a cliche, but fail early, fail cheap is the motto of successful biotech outfits. And then just to say, the regulatory pathways for gene therapies are still evolving. They're not locked down. There isn't a fixed guidance to follow, but they are established. There's a process. We know what the expectations would be for a given therapeutic. We can actually plan ahead and look ahead and think about, this is what we need to think about and manage in terms of getting the best interaction out of the FDA and making sure that programs stay on track and don't get unexpected holdups. And then lastly, just to say, and again, the complexities of these biological structures that are gene therapies does mean that you do need an early engagement around CMC and manufacturing and GMP manufacturing, and actually getting that clinical process early to understand how that's going to work and how you're going to be able to develop to the clinic. So I think I've hopefully managed to keep reasonably well to time, and I'll see if there are any questions. Thank you.
Thank you so much, Keith. That was incredibly informative, and I always reminded of the adage that the more I learn, the more I realize how little I know. I think demystifying the gene therapy process for me has been, over the course of the years, learning how complicated it can be. And I'm hoping that some of our listeners today really kind of understand that we are that we're in good hands, and B, how complicated this is in the myriad ways in which it can go south. To that point, I just want to talk to you, but while people are thinking of questions, that this, when I introduced you, I mentioned that you'd worked with hundreds of gene therapies, and that is correct statement. Is that not right?
Yeah, so Charles River is an organization. I forget, we've across multiple programs. I mean, we as I say, we support all sorts of sizes of companies and at different stages. We support in early discovery, in that preclinical, most of what we do is preclinical safety assessment. We also support at the clinical stage. So as an organization, we see the breadth. I mean, I think the people who truly see the breadth are the reviewers at the FDA. And I think that's the bit that they're actually one of the most important people to have honest conversations with to guide your program. But within Charles River, we see that we have, I mean, I'm one of many people with expertise. And in terms of our developmental expertise, we have, so in terms of regulatory side of it, we've Lauren Black, she's an ex-CBER reviewer, so she actually comes in from the FDA. We also have a colleague actually of mine in the UK, Helen Marie. She's formerly of the MHRA, so the UK equivalent of the FDA. So she was a clinical stage reviewer for them with a lot of familiarity and experience. So we have that kind of execution level experience, and within that we often work with clients. When we broadly work with clients when they're talking about how they're presenting to the FDA or they're coming to us, we've got this feedback. How do we interpret? How do we work for that? So our role is always to help, to support, and it's always focused on shortening that time span and having those conversations with clients about how do you stay on the tracks. Let's stay on the tracks. There's always something shiny, especially I think when you're working with academic spin-outs. I mean, they're the best clients for me to work with because they're the most engaging. But again, there's often that bit about, this is a translational journey. You can see that over there, and it's really interesting, but these are the tracks you've got to stay on to get to the clinic.
And I think that's, when we talk about biotechs a lot, whenever there's a new patient diagnosed or people come forward and they want to know more about this, the first question is, well, this company is doing, Pfizer or dentists or Sarepta, they're doing this in muscle already. Shouldn't we be going to them? And of course we are in discussions with them, but what I want to reiterate for our audience is that you know what they know and then some, because you've seen it from so many different angles in so many different trials, and you sort of understand the pitfalls that others have had to, that we can now avoid thanks to the lessons you've learned through them. Is that correct?
And that's correct. I mean, and I think we obviously, to answer, we absolutely respect client confidentiality. So there are levels in there about what we can and can't do in terms of what we do. But equally, if we know that what a client is proposing has never been asked for by the FDA, we'll have that conversation with them. Why are you doing that? Who told you to do this? I mean, we will try and have those hard conversations with clients. Sometimes we succeed, sometimes we don't succeed. Equally, when a client sometimes come to us and they've had comments from the FDA and they're going, well, what does that mean? We're saying, well, maybe it means this, maybe it means it, but we can give you an educated interpretation of, based on our prior experience. I think that's where they're kind of trying to push you towards and to what you should be thinking about.
I think that's valuable. And in terms of those hundreds of gene therapies, have they mostly been in either children or small confined areas? Because I know you mentioned toxicology and the risks of it. And I know that when you do a gene therapy in a small organ or, the eye or a baby, it's going to be different in terms of results than in, say, a 150-pound adult. What is that in stone? I'm sorry, but I'm sure you get size adults. So what are the risk factors there, and what are the challenges there?
So the first challenge, and this was the point I was making about when you design a program, you're designing it for a clinical population. So if you're designing a program for a pediatric indication, that's going to look very different for a program for an adult population. Those are two totally different programs. The risks, I mean, all the regulators, it's a risk-benefit calculation. So if you're looking at children who have, a standard of care or a reasonable prognosis, that's a very challenging group to target with the gene therapy or any therapeutic, because there's a sensitivity about what happens if you get that wrong. Also, this year under 4%. If you're working with adult groups, you can have different. And so the programs look different, and that also impacts how you administer a drug. So one of the key aspects of the safety phase is we have to administer the drug as you will in the clinic. So that's where, so programs become very different if you're looking at an intravenous and an infusion administration. So for example, if you're targeting the skeletal muscle, that's an IV infusion generally in terms of administration. That's a very straightforward, well-established process. That's okay. If you're going in for a gene therapy to treat a neurological disorder, and you basically have to use stereotactic guidance to deliver to specific locations within the brain, that means your safety assessment phase has to capture all that. And so it becomes much more complicated. So this is this whole question about, is there a standard approach? And it's not, it's entirely driven by what it is you're doing in the clinic and who you're treating in the clinic, and what their standard of care is. The regulators are very attuned to risk. It is risk and benefit. How is that going to be captured? How do they assess that?
Right. And with muscle, given that it's sort of systemic from the top of our head to the bottom of our toes, that drives a different kind of an approach. And I suppose that's tied to what kind of AAVs we use. What thoughts do you have on that, and what experience do you guys have with that?
So the standard AAV for muscle programs, so the programs that are in clinic for Duchenne, tend to be AAV9. Because that, we all know, that's well established to reach the muscle. There's obviously, there are a lot of companies out there who are looking and trying to finesse the design of the AAV capsid to kind of improve the targeting, because one of the limitations of AAVs is AAV9 is good for muscle, better than say an AAV4 or an AAV5, but that's not absolute. So you can't say, I want to hit the muscle, that's an AAV9, I want to hit the liver, that's an AAV4. They all hit these kind of overlapping sets of organ systems. The bias is more towards one, so AAV9 biases towards muscle, but it is going to go elsewhere. And so that's, again, something one needs to think about and assess in a program.
Because the NDF, we are funding also in parallel with all these efforts. We are funding next generation AAVs. So I think that's a key point. Thank you for bringing that up. And then I have a question here from Dr. Monkolek. And he says that one of the biggest challenges in therapy development is costs. Don't we know that?
Yeah.
Do you anticipate any technologies or regulatory workflows in the near future that may help in reducing this cost?
So for gene therapies, I think one of the major cost impacts is actually the manufacture of the drug, the CMC, the drug manufacturer aspect is massively expensive, and it can actually cost more than the safety assessment. You can probably add up some of the costs on a lean program. Even though you're spending a lot, if you like, on that proof of concept and that safety assessment phase, the pharmacology and the safety assessment phase, the bill for producing your clinical material can be more than that. And I think that's probably, as we're going forward, the area hopefully we're going to start to see. We've got more, there's more and more. Charles River are now involved in that space and things, and there are a lot more companies pushing innovative technologies and platforms to try and manage that. But there is a basic truth. I think this is why I wanted to touch on CMC in my talk, because of the real complexities of the therapeutic, and hopefully, and I would be confident, because the one thing hopefully we can all rely on is progress, even though sometimes it might not feel that way.
Right.
That aspect will,
Listen, we have a saying here at NDF that time is muscle. And so I know that is definitely on everyone's mind, in terms of it. But I believe there is progress. I've seen leaps and bounds in the last few years that we've been working towards this. I believe we've really been in a good position of duplicating the things that have worked and avoiding the pitfalls that others have experienced that have gone on ahead of us. And now with your leadership and guidance, I think we are in really good hands to make sure that we can dodge and weave as best we can. But I do believe on a good note, and I'd like to end this call on a good note, is that having a background in Silicon Valley, and I don't even think you need that to know that technology, just the way it works, is that the longer it's around, the better it works. And that does in fact drive prices down, so we don't all have to SMA, what was it, $2 million per dose for a baby? I think that's going to be a thing of the past.
And I think, and that is, these are the aspects and the challenges, and I think there will, as you do things, the first time's the hardest time, the most expensive time as you go through that. And I think this is one of the things that, as you come to that, you kind of think, starting now where you are, I mean, we know a lot, the world has moved on a lot in the development of these therapeutics, and we can focus down programs and say, because the FDA, the regulators are getting much clearer about what they need and what they want, we can think more clearly about how we get to candidate as quickly as possible, and all the ways you can think of just cutting that decision-making time and really being harsh. I think one of the key things is to be really harsh with a program and say, look, because I mean, one of the truths for the large pharmaceutical company is when you pay for a drug, you pay for the failures.
Right.
And that's how, that's how it has to be. We have to pay for the failures. Right. But there is that bit about, well, if you are going to have failures, fail early, fail cheap. It is a cliche, but I think it really does matter in drug development, because it gives you time. I mean, if you save the money, you get the time to go back.
Exactly. No, not.
Yeah.
Right. Right. I think that's amazing. And then on the final question that I think everyone is thinking, no one's typing because they've asked it so often, is what, in terms of timeline, in terms of GNE myopathy, and I know we've discussed this in the past, specific to our disease, the road we're on right now that was architected in part by Dr. Lack, Angela Lack, and Monkol Lack, with the support of Rich Horgan and some of our other scientific advisors. We sort of feel comfortable in that 18-month to 2-year time frame where we could actually begin dosing.
And I think that's I think that is a reasonable goal, and I think you have to have that as a goal and that kind of plan for success attitude. And I think you do need to have that time, like that time goal. And again, it's to think biotech, like, drive those goals, drive those mileposts, and say this is when, because they are achievable. But they might be hard to achieve, but I think that's things which that is something that can be done.
So that's really good news, because the sort of more academic way, the more traditional way, we had a timeline of about 5 years. So for those of you listening, we've with the support of this amazing CRO, the folks at Charles River, we've been able to shave off potentially 3 years, at least in theory. And I think it's been done before, so it's not going to be a big surprise when it happens for us.
This is important. And I think one of the challenges sometimes, I think at the academic level, I mean, I think there can be a quest for the perfect drug. And that's understandable. You want to, can you tweak this? Can you improve it? Can you get a little bit more? But eventually you've got to stop that. You've got to make, and this is a bit about go, no go decisions. Is this credible? Do we honestly, hand on heart, believe if we put this in a patient, we would see a therapeutic benefit? And I think and this is a bit about being honest about things, plus good and bad. Is that something that if we delivered to a patient, if we put it on that, we would be confident we'd see benefit?
Thank you for saying that. I've had this discussion over the years with a lot of people who have wanted to push us to go faster. And I have to put my hand on my heart and say, I swear to you, until such time that I would do this to my own daughter, I will not allow something that is not potentially safe. And that is the question each of us should be asking ourselves. Is this something that we would recommend for someone that we love and care about, which is, all of our patients? And until that point, it's got to be a no.
You don't. And I think that is the bit. I mean, I think it is this bit about just developing something for the sake of developing it isn't, because actually you'll get to the FDA and they'll just
they'll shoot it down. Yeah, and that's the other thing. It's not about scientific curiosity or keeping labs going. This is really about coming up with solutions for people who are in tremendous difficulty. So having said that, Dr. Sutton, I wanted to thank you so much for your wonderful presentation. I know this will be downloaded many, many, many times, and we look forward to working with you moving forward. So
I look forward to it, and thank you for the opportunity to present. And I too look forward to working with you over the next few years. And
wonderful. Thank you, everyone.
Progress.
Talk to you all soon. Bye-bye.
Bye-bye.